Okay, I'd like to start off with our case study on twins and identical fates. It's a study that's supposed to introduce you to this idea of epigenetics, and we'll get into that in just a moment. Your questioning activity that I gave you earlier in the week, if you remember, had to do with this particular section of the case study, where you read the case study and you answered certain questions, and you were supposed to come up with questions on the passage. You didn't have these questions. You just had the passage coming home. You were supposed to come up with questions you would ask. For instance, if you are a a physician or a counselor and you had this case in front of you, what kinds of questions might you ask about to try to understand the case a little better? Some of you asked really, really great questions. I've read some of your questions already. I will continue to read them throughout next week. But when we look at these questions, you'll see what exactly are twins and how do they arise. Your response should distinguish between the two different types of twins. Now, that answer I've given you in class. It should be in your notes already several times in this quarter, in this year. But if it's something that you don't understand and you don't have in your notes, this is something you could put into Google and find the answer to. You'll see here that these two twins are identical twins. So that gives you some information that you can work with because of this passage here that says identical twins. So when you're looking at the passage and you're trying to find out what exactly is going on in any part of this, you will find key ideas and terms within the passage that's going to help you answer the questions in the case study. Are identical twins completely identical? Why or why not? This is information that you have to look up. You should be able to Google it and try to figure it out. And what can identifying twins tell us about genetic influence on a particular trait? What can studying twins tell us? So when you look, to help you out with some of this, I'm going to go ahead and give you a broad answer. You're going to need to look up some specifics on Google or in your textbook. And so one of the things that you have to think about is so after you've identified twins and the different types of twins, and again, you should look at your notes and go online if need be. The next two questions really have to do with these two topics that I put in these boxes. The first, are identical twins completely identical, why or why not? That has to do with the nature versus nurture debate. And that's how much of you is your genes and the result of your genes being the way they are. And how much of you is how you're brought up and the choices you make in your life. So the nature bit is the stuff that you inherit from your parents, and that's outside of anyone's real control. You are who you are. You get the genes that you get, and we've been talking about that through studying that in Punnett squares and talking about it in the sense of the different kinds of genes and how they produce their gene products or proteins, and how they work and interact with each other, determine how we see those genes, those two alleles once we've started to develop. So the nature versus nurture. So in nature, we're talking about which alleles did you get and what's the result of those two alleles. Some real simple stuff that is beyond debate as far as nature is whether you have sickle cell disease or not, whether you have any kind of genetic disease or not. That is 100% nature. Now, what's interesting is even that there's some nurture does have an impact on that inheritance of those particular genes. For instance, not all people with sickle cell disease, even though they all have the same genes for sickle cell, not everyone suffers in the same way. So your choices can influence the outcome even if you do happen to have a genetic disease. Sometimes they can't. Sometimes the genes, for whatever reason, whether it's the version of the disease gene or a combination of other genes with that particular gene, sometimes that overcomes any nurturing that happens, anything that you can do with your environment, for instance, eating low-fat diets or drinking excess a lot of water, exercising. Sometimes those things don't help at all, even though you do them, they just don't help. We know, for instance, in a recent study that cancer is pretty much a random event. So even though the environment has very little to do with the outcome of your cancer, whether you get cancer or not. Now, it's arguable whether the outcome, whether the fact that you die from cancer, survive cancer, or what have you, does the environment, the foods you take in, the vitamin supplements, the air you breathe, the stress level in your that you live in, does that influence the outcome of receiving a certain set of genes? That's still up for debate, and every situation is going to be different. But the fact that you inherit a particular set of genes and that some outcome happens, at least at a preliminary level or at a base level, then we'd have to say that that is definitely nature. Okay. Now, the environment, even though you have a good set of genes, your environment, whether it be the way you were brought up or the way what you eat or what you choose to do, your lack of exercise, that's going to impact your outcome as well. And you might have the genes that would let you live 200 years into the future. But your choices, your life choices, your eating habits, your exercise habits, they could influence your lifespan. And we call that nurture. All right. So it's always nature versus nurture. This argument, this kind of back and forth discussion, these considerations always come into play when you're trying to look at two people and whether they're identical or not. So you can get two twins that were raised in different households. One's fat, one isn't. One has heart disease, one doesn't, even though they both have the same genes. But assuming a similar upbringing and same genes, again, there's this idea that if this is the case, then you're probably looking at two very identical human beings. Now, since identical twins have exactly the same DNA sequences, and because they result from an early mitotic split, so early on in the development of the fetus, there was a split, a mitotic split. Remember, mitosis gives you an exact copy. That is why you have two separate human beings with exactly the same DNA, because of that mitotic split. That was after meiosis, right? After you have conception, some mitotic split happens and you get two fetuses developing with exactly the same DNA. That is what we call identical twins. You'll have to look up the other types of twins and how they happen. Again, look in your notes because I discussed it. Are there any different, you have to consider any differences between the two often are thought to come from the environment. So what part of a disease, for instance, if one twin has heart disease and one doesn't, and they have identical genes, now we are pretty sure, we're almost 100% sure that it's not the genes, but the environment, the eating habits, the lack of exercise or whatever that's actually causing the heart disease in one at an earlier age than the other. The same thing goes with a lot of different diseases. So when we're looking at this, we're looking at the difference between twins. That's why twin studies are so important, is because they help us understand the disease pathway. They help us understand the causes of the disease. Some people, for instance, if you have sickle cell disease, you have sickle cell disease. You have the two genes for it. But one twin might have a less stressful life, might live longer than the other twin. The difference between those two individuals then is thought to be an environmental difference, and that could help us understand what kind of environment do we have to foster in a person with sickle cell disease that would help them live a longer, healthier life. So twin studies are very helpful. Now, I want you to go back and look at these three questions and make sure that you can answer them if you haven't answered them already. Anything you still don't understand, Google it or ask it in class after school or during our Saturday sessions. Remember, next Saturday, I know this Saturday is Our school's closed, but next Saturday the school will be open. So consider that as we move forward. Okay, in the second part of the diagnosis in the second part of the case study, we find out that Elise discovered that her sister had schizophrenia. And Shannon, the girl that her sister, the one that was diagnosed with schizophrenia, um, had shown signs of it, and you can see that in the first two paragraphs. You also see that Elise, in her research, found that that schizophrenia is inherited in families, and you see it has a tendency to. See that it has a tendency to run through in families, and the fact that studies indicate that sibling schizophrenia is 10 times or tenfold higher risk of developing schizophrenia than over the general population. So that's these are important facts to understand and why Elise is worried about getting this disease and this disease of the mind. Okay, the next three questions have you looking at these articles. Now, you can look these articles up, or you can just find a Google site or Google Scholar article that discusses the same topic. You'll see here this question: what causes genetic variation? What causes some people to have curly hair and others not? You should know this by now, right? And we've discussed it, and you've been tested on it. Certainly you should understand by now that meiosis is sex is what is the source of genetic variation. Certainly there's some mutation involved, but in general it's crossing over, it's independent assortment. And this is what causes people to get different genes and a different combination of their genes, even more with a higher degree difference than just sex alone. Meiosis, crossing over in meiosis, and getting half your genes from one parent, half from the other, and then crossing over, and then independently assorting these chromosomes gives you this higher degree of genetic variation, where mitosis, you're getting exactly the same genes. So you see this question, the answer to this question is exactly what you would expect it to be based on what we've covered in class. What causes some people to have genetic disease, such as cystic fibrosis, and others not to? Well, I think you should understand that by now, that here we're talking about that person inherited a gene or set of genes, right, depending if it's a recessive or a dominant disease, that means that person is going to develop a disease like cystic fibrosis. Now, so that being said, I suggested to you in the first in part one that some genetic diseases might be made better via the environment. So even though you inherit a disease, does it mean that it's going to be as severe as someone else's? And can we influence that disease with the environment by changing our behavior or changing where we live or what we're exposed to? That may be true. Again, it's dependent on the disease and it's dependent on the individual. So, but the bottom line is that the cause of the genetic disease is a gene or a set of genes, dependent if it's dominant or recessive, that determines if you get a particular disease or not. I used sickle cell anemia a minute ago. In this question, they ask about cystic fibrosis. It's the same concept. You're either inheriting one gene if it's dominant, two genes if it's recessive, and therefore getting that particular disease. Now, what does it mean to have a trait or disease that runs in families? Well, here again is the idea that things are inheritable. So genes, the trait is what we see, but the genes for that trait, the genotype, is what we get from our parents. So of course you'd expect them to run in families. So you'd expect that if you have a gene, that those genes, this trait is gonna run in a family, because one parent's going to give it to the child and the child's going to give it to their child. And so you'd expect the disease to show up more often in that family than a family who doesn't have that particular disease, right? Those particular genes. Now you might ask, well, how does that family get it? Well, they marry someone who has that gene. it's the only way to get it unless there's some kind of mutation that occurs and that causes it as well. A novel mutation that causes a disease is not as likely as some parent or someone marrying into the family and creating an increase in the particular disease or trait. Okay, so what does it mean to have something run in a family? It means that there's a gene that has been inherited from an ancestor that someone in the family probably has, and it will continue to turn as long as they have babies. Will continue to pass it on. So, what could some possible genetic and non-genetic causes of Shannon's schizophrenia? Well, we talked about genetic causes. It's genes that are inherited, and you can look up the specifics in these articles if you can get a hold of them, or you can look them up on Google Scholar, or even just Google and look up schizophrenia and what are the causes, genetic links, and what are the environmental causes. Now, there's a lot of different types of schizophrenia, and you could spend a whole year learning about just this one disease. But there's things like schizophrenic episodes the schizophrenic splits can happen because of the environment, because of stress, because of exposure to a chemical, any kind of a multitude of reasons why someone would develop schizophrenia. I don't think it's a disease that's well understood yet. I think if you look at some genetic disease, you inherited a disease that makes you more likely to get it. And so what might, if there's some environmental influence, for instance, some kind of insulin shock or some kind of stress that her sister went through, or some lifestyle that her sister, drugs, for instance, that her sister might be doing that she doesn't, then maybe Shannon's schizophrenia will not show up in Elise's life. So, drug use is one thing that I know that can influence the development of something like schizophrenia. All right. Well, in this particular part of the case study, you start to look at more biochemical results or molecular reasons for particular diseases, for someone to have, for one twin to have a disease while the other one doesn't. And one of the things you note that you'll see here in this particular section of the case study is information like schizophrenia is almost 50% inheritable, which means that about 50% of the cause of the disease is a gene or an allele, while the other 50% is due to the environment. So again, this is that it's a complex disease, as they lay here, and so are many diseases, genetic diseases. And 50% of the cause is due to other things. So while 50% is due to alleles, 50% is due to things other than your DNA. That being said, even the 50% that is due to your genes, all right, that's the part that the two sisters share, their genes, that the environment can actually influence the gene. This is something called epigenetics. Now, this is something that's relatively new as a study. We've known for a while that genes can be turned off and on. In fact, I learned that when I was even in high school. So at the molecular level, we knew that genes had to be able to be turned on and off. But it turns out that the genes and the things that they produce, the proteins, can be impacted by markers, by how they're packed away and how they're unrolled. Now, one of the questions here, describe what you know about the structure of DNA and how DNA is packaged in a cell. Now, you know that when genes are being used, you have the form called chromatin, so the DNA is all unwound, and you know that a chromosome, as defined, as strictly defined, is a compacted DNA. So a DNA that's wound up on histones and then put in a particular manner so that they form the shape that we're all so very, very used to, which looks something like this, with a centromere connecting it. Of course, these two being sister chromatids, right? And so, but this is what we would normally have considered a chromosome, and that in this chromosome there are these things called genes, and then these genes exist in a specific locus, right? And we know that. And so we have one gene in one place, another gene somewhere else, and but the location of each particular gene is set. That's because that's the DNA sequence, and that sequence doesn't really change. Well, it turns out that during packaging, there are certain chemical changes, changes that can be inherited. So it's not just temporarily turning something off because of a particular environment, like for instance, a cell turns off some genes to and turns on others to become an eye, and another cell turns off some genes and turns on others to become a finger. It's not just that, because these genes can easily revert back with using some molecular techniques. We can revert these back to being able to produce any one of the tissue types that make up your body. But it turns out that these genes, after you make an eye, after you make a finger, then these genes can be further changed through things like adding these little groups called methyl groups. And they look something like this. So they could be attached to any one of these nucleotides, and then that changes, or the histones, the proteins that they're wrapped around, and that changes how these genes are expressed. And so that could then, if someone has a gene here, let's say these two sisters share this gene, and if it's methylated or if it's changed in some other way, that then activates a series of biochemical steps that leads to something called schizophrenia, or could lead to something, to any number of diseases. Then we can get one twin which has the same DNA sequence, but then has lived in an environment or has been exposed to environmental factors that cause her DNA to be methylated or somehow changed. That twin could get the disease while the other one doesn't. Or the opposite could be true, right? So the person could have be destined to get schizophrenia, but something happens. They do something. They're exposed to some element or some molecule or some UV light, or they swim in the right ocean or something. They're exposed to some environmental factor that methylates it, changes that gene, so that person doesn't get it, and although they might have gotten it. So the fact that you have sisters or brothers or even twins that share DNA, that share genes, does not destine one to get a particular result, a product. Things can be changed. Things can be inherited differently. Changes can happen after you've inherited the gene that make that product, that person, develop some disease or some good can come from it that will be different from the person they share that gene with. So genes are not the whole answer. They're only part of the answer. Again, that whole nature versus nurture, the environment versus your genes. So that's the structure of DNA, and of course we're going to go into this in chapter 7. That's what we're starting when we get back together. We're going to get into DNA and its structure, and exactly what does it mean to have epigenetic changes to your DNA. Now, the next question is, at the molecular level, speculate some ways the environment might have influence on the DNA and its packaging. I just gave you some examples, and methylation of a histone. Remember, the histones are the proteins that the DNA is wrapped around, and we know that DNA is wrapped around histones when you're doing mitosis. So you can see clearly that when you're dealing with DNA, there's all these very finite, very, very complicated steps that can make a single gene produce a disease or produce a positive influence that someone with the same gene may not have. So that kind of explains why two people with the same genes, whether they be twins, whether they be related or not, two people can have the same genes and one person can get the disease and the other not. So it's very hard when people say, so-and-so, you're increasing your risk. You are increasing your risks when you're doing any particular environmental action, any decision you make. You may be increasing or decreasing your risk. but it all depends on your genes as well. So there's this play between your genes and the biochemical changes that are occurring in your genes and what the outcome is in the end. So that explains again why the environment, what the environment has to do with and how with DNA and how it influences DNA and its packaging. We just discussed it, and I hope you understood the general idea. We'll discuss it more as we get into chapter 7. This all leads us to part 4, which is really, really cool. So, here in the story, Elise goes on and she learns in her intro biology class, her general biology class. You can see that's listed here, right? Her general biology class. And she learns from Dr. O'Brien that there's this stuff called epigenetics, which I kind of mentioned earlier in in part 3. And they talk about this modification called DNA methylation, which I discussed again in section 3. And they talk specifically. You can see that this is one of the bases, right? One of the DNA bases that you find in a double helix. So if you remember, a double helix looks something like this. And then one of those bases could be, it could be this cytosine here. This is cytosine, one of the bases in your DNA. In other words, it's the C that everybody likes. It. We always say A. It binds to T and C binds to G. And this C is called cytosine. All right. Again, we'll get into that in chapter 7. But what's cool here is that the process of methylation, or the adding of this methyl group here, all right, that's been pretty well described. And so you have this enzyme that takes the SAM molecule, takes the methyl group from the SAM molecule and attaches it to the cytosine at this position. You'll note that before it had just an H on it at that position. Now you have a CH3 or methyl group. That changes how the DNA is read. So that changing of the DNA and how it's read and how it produces a product and how you get a result could mean the difference between someone getting a disease, having a disease, or not having a disease, someone living longer, someone digesting a food better, someone seeing better, someone thinking better. I mean, all kinds of things could be different. And again, this methylation happened because of the environment. It happened because of the environment. And that's really important to understand, that the environment now can actually influence an inheritable change in the DNA. That the change in the DNA caused by something someone's eating, or exposure to sunlight, or exercise, or exposure to a toxin, that this stuff can stimulate a methylation to a DNA molecule that then changes how that DNA is expressed. A very, very powerful idea that we'll talk about more in chapter 7. But here you have the twin study results. You've seen these southerns and northerns and westerns in Mr. DeCipeda's class. You've talked about how gel electrophoresis can be done to separate molecules like DNA into sections. And this is a twin that has no methylation. So the changes here that you see going on in here did not happen to this twin. This twin might have married someone and moved to the south and it's been exposed to sunlight. Or maybe this twin here was exposed to sunlight. Whatever environmental difference between these two twins that caused this change to occur. And here are the 3 spots, here, 4 spots, here, and here, that you get methylation occurring. So this result shows you that at the 3-year-old, this is the DNA at the 3-year-olds for twin A and versus twin B, but this is at 50 years old. You see that there's a difference. This twin B has methylation here, so you don't see that band here. You see there's methylation here. There's no band here. This twin had a methylation that this twin didn't. And this twin had, again, 3 methylations now. 1, 2, 3, that this twin did not. 1, 2, and 3. They don't have that methyl group. Where this twin here has a band, a methyl group, that this twin doesn't. You notice that when they started life at 3 years old, they had the same exact sequence. Once they've grown older, different genes have been methylated in different ways, again due to their environment. Now, these two twins are still very much alike, much closer, much more alike than any one of us at random might be, and any two humans at random would be alike. But they're slightly different because of how they lived their environment. And that nurture then influenced their nature. Their environment influenced their DNA. Very, very powerful idea. All right, then this takes it to the next level and it starts to look at some using labeled chromosomes found at taken from the twins at a normal metaphase stage. Now we know that at metaphase we're talking about the sister chromatids starting to line up. So we can take a look at these metaphase chromosomes and separate them. We have the technology to do that, and then label them with a fluorescent molecule, and we can label the non-methylated DNA in green, and you'll see here that anywhere you see green, that DNA has not been methylated, while where you see red, that particular DNA has been methylated. And wherever you see the yellow, that DNA has equal methylation to non-methylated, so equal red and green. And you could read this section here if you really want to know the details. It basically says exactly what I just said. Now, where it says monozygotic twins, that means that the twins result from one zygote, and so they're identical. Monozygotic twins is the official way of saying identical twins. And hybridization for methylated DNA, we're talking about that hybridization just means that the pigment, the fluorescent molecule, has bonded to the methylated DNA. And you have you see here 4 representative chromosomes. There's 2 here from twin 1 and then twin 2. There's 2 here from twin 1 and twin 2. This column here is looking at 3-year-old the twins while they're 3 years old, and this column is looking at the twins after they're 50 years old. Now, if you notice that the pattern of methylation versus non-methylation is pretty much identical between these two. There's slight small differences, but for the most part identical. And it's true throughout all 4 of the chromosomes that we're looking at. Each one of these is a different chromosome. They don't mention which chromosome, but let's say chromosome 1, 2, 3, and 4, just for argument's sake. And you see that the patterns look exactly alike in between one twin and the other twin. Fast forward to 50 years. Fast forward about 50 more years. It's more like 47, but whatever, 50 years. You'll notice that the patterns are different. There's not a huge difference, but there are differences, much bigger differences than they started with. There's a lot of methylation here on this twin, for instance, and not so much here. You see this region here has a little more methylation. This region here has a little more methylation than this region. This whole section up here is methylated, where this one's less so. This one looks like it's methylated here and here. You'll notice that there's a lot more about the same amount of methylation at this spot. Here they're slightly different. And this chromosome has more methylation, for instance, than this chromosome in general. So there are going to be some differences there. You'll see here that this chromosome has a lot more methylation here than here, and a lot more methylation here than here. And finally, this chromosome has a lot more methylation here than here, and a lot more methylation here than here. So the point is that there's a lot of differences, a lot more difference, a lot greater difference in between these two sets of DNAs after 50 years of living, of different exposures or what have you, than after only 3 years of living. Okay, so the methylation is increased, and where that methylation is differentiated, it's different. That's a really, really important concept, because that means, this twin might live longer because of it, or maybe this twin lives a better life. This twin gets a disease because of the methylation here. This one doesn't. They have the same basic DNA. It's just that one's been methylated and the other hasn't. There are other kinds of changes, other kinds of epigenetic changes that can happen. We're just looking at methylation here. And so when you're looking at the data shown in these two, they ask you here which set of twins, 3-year-olds or 50-year-olds, have the most similar DNA methylation profiles. I think you see by what I just said a moment ago that these sets of 3-year-olds obviously have a better or more closely related methylation profile than these two. And the explanation I just gave, I'm not going to repeat it. So you could just tell, they say if you read here, you can see the red. And why would that be? Well, that would be because these twins have lived about 50 years in different environments, have made different choices to drink or not to drink, to smoke or not to smoke, to do drugs or not to do drugs, to eat high fat or not eat high fat, to exercise or not exercise. All the, to live in the north versus the south, to travel or not travel. These are, one may have caught a disease, a virus that has triggered some methylation, or, there's all kinds of things that can happen. And then that goes down to the answers question 2, and you can look up more of what influences DNA methylation. But we're just starting, really. This is a relatively new science, so there's a lot of study that still needs to be done to understand the influences of DNA methylation. Now you can also hear, I want you to go ahead and look this up. What other types of epigenetic modifications can occur within a genome to influence gene expression? You go ahead and look those up. There are numerous ones that we've identified so far. By a long shot, we probably have not identified every different type of epigenetic change that can happen between two twins. Now imagine all these changes that we just talked about between these two twins at 3 years old and at 50 years old. These two twins, the changes that have occurred are only due to methylation. Now imagine the other epigenetic changes that could have happened, again because of all those environmental influences, that would make these two twins even that much more different. So as twins grow older, their DNA literally changes, and they change in different ways, so they become less and less alike. And that's true of all of us. All our DNAs are methylated, and so our DNA changes over time. So it's not just our thinking and our body. It's not just hormones. It's just not menopause, or it's not just getting fat, or not exercising enough. It's also the DNA itself starts to change because of these environmental influences. And do you think Elise needs to be worried about her own mental health? Why or why not? If you were a health professional, what would you advise Elise to do? I almost want to let you answer this, but because I just want you to focus on looking up just one set of information, which is what kinds of or what types of epigenetic modifications can occur. I want you to look that up and make sure you put that answer in. I'll go ahead and give you this one, and since it's, I think, relatively easy to think about if you give it some thought. And that said, yeah, she should be worried slightly. I mean, she's still a twin, right? So she still has the same genes as her sister. She may even if the schizophrenia is caused by methylation of a particular gene, that methylation can occur to her gene if she's exposed to the same environment. If I was a health professional, what would I tell her to do? I would, by the way, I certainly wouldn't be as worried as if this disease were 100% due to a gene, right? So it's only 50% due to a gene, the other 50% due to the environment. And again, the other issue that we know is methylation also changes it. So it's really two different people here. But what would I advise her to do? Certainly to keep an eye on her own personality, to keep a diary, to talk to her friends. And if she starts hearing from her friends that there's some changes going on, she should seek professional help and identify if this disease is actually starting to develop in her, because this disease is treatable. So she should keep an eye on it. So there's a lot of antipsychotic drugs out there, and if she ends up getting the disease, she can improve her situation. At this point, if you've completed all the other parts 1 through 4, you've done it well, and I grade it, and it's an A. Part 5 is extra. It's a little deeper. It takes a little more thought and really has you investigating some complex biology. If you're willing to give it a shot, that's great, and we'll call it extra credit. And I think it's important, on this particular activity, we'll go ahead and give it 100 extra credit points for the activity, not just extra credit points, above and beyond points. So when you look at this particular one, again, if you're done, if you've completed, you don't want to go on, you don't have to watch the rest of this video. If you're interested in getting those extra credit points and really trying to understand some deeper, higher level biology, let's go ahead and continue having this discussion. Your choice. So here, there's this protein called reelin, and it turns out that reelin is a glycoprotein, so it's partly protein that has some sugars bound to it. And it bonds to these intermembrane proteins. Now, this is the membrane of the cell drawn as a line. Here's this protein sticking out. There's a receptor here, and this is the protein that is binds to that receptor. Let's take a closer look at that. And you see here that by taking a closer look, you see here that this particular receptor, this protein binds to this receptor, and through a pathway, so this changes the shape of this receptor when it binds, which changes this protein, which allows it to be phosphorylated. There's a phosphate group binding to it. We've seen that before, haven't we? And then that particular change goes down and influences some other proteins that finally do this. They have neuron migration, they have brain development, learning and memory, and synaptic transmission. So this protein actually influences your brain and how it thinks. So it's easy to see that abnormal methylation here could cause the brain to develop abnormally and develop something called schizophrenia. So in the story, as we move forward, this Dr. O'Brien is going to continue to study this epigenetics and consider what influence does epigenetics have on the development of brain activity and therefore schizophrenia. So he does some studies, and let's go ahead and take a look at these studies. Here is done on the prefrontal cortex layer, right? That's the part of the brain that does the thinking, right? The imagination. Now, I want to point out something for those of you that have stuck with us. Here's this little bar. It's an error bar, so it tells you that this is true up within this range of error. And so you see that bar in each of these data results. And you see that the non-schizophrenic subjects are here, and the schizophrenic patients have this level of that particular protein, right? So as we go through the layers of the prefrontal cortex, you see that the reelin is in higher concentration in layer 1 and much less concentration in layer 6. And in fact, you see that schizophrenics have about the same amount of reelin as the non-schizophrenic subjects. But in layers 1 and 2, the difference in reelin concentration in the particular neurons is quite striking. I think this one's about double. The non-schizophrenics are about double the reelin protein concentration in the protein or reelin positive neurons. And here again, it's about double, if not more. The difference isn't even more. I think here we're at 15, and this one's at I want to say 35. So it's even more than double the amount of reelin positive proteins. Where here in the third layer, they're much closer together. So this kind of graphic analysis, by the way, is what you're going to have to do on your PARCC exam, whether you're doing, your physical science, your social studies, your English, or your math. They're going to give you a lot of graphs to try to analyze, and so you have to become familiar with trying to analyze these particular graphs. So here, if you're just looking at it, one would think, well, it looks like reelin's important, especially in the first two layers of the prefrontal cortex. Okay, so here in this particular graphs, these two graphs are three actually, we look at the methylation of non-psychotic patients after postmortem, after they've died. They've done an autopsy and they've collected the data, And here we have percent of methylation per site. Here for non-schizophrenics, that's all along here. And these are sites. These are positions of bases in a chromosome. And here you have all these are positions of methylation, percent methylation within. So this particular position, you have something like 32% methylation, where at this one you're looking at probably something like 5, maybe 3% methylation, where this one here is 10% methylation at this position. Now you look at schizophrenic methylation, percent methylation at the exact same positions. You notice that the positions are the same. And what do you notice? I'll give you a second to think about it. Well, I hope you notice that at position negative 135, you have a lot more methylation, and we would even call that abnormal methylation. Remember, this is your control group. It's like any good science, you need a control group. You need something to compare it to. Otherwise, how do I know that this is abnormal? I don't know that any of this means anything without a control group. So you need a control group to compare it to. So here's your control group, and you see that this methylation here is extreme. Extreme. In fact, overall, there's a lot more methylation across the board. But at this negative 135 and at position, what looks like 20 or something like that, you have a lot more methylation than you do anywhere else in the normal range. And in fact, even this position here seems to have at negative 365 or so, you have a lot more methylation here. It looks like 28 or something like that, where in the normal it was something like 18. Is that significant? Well, that's up for debate. But the bottom line is that in each of these positions, you have a lot more methylation than you would expect in a normal patient. There seems to be a strict correlation here. Is it a cause and effect? We don't know, but it certainly is. It certainly is a difference that we need to consider. Now, we also know the biochemistry of it from previous studies, right? So we know that methylation can cause a change in the pathway that leads to normal neuronal function. We looked at that at the previous page, the previous slide. And here you see that there's definitely a difference in the number of renin positive cells in schizophrenic versus non-schizophrenic. And here we actually know the positions. Now, what would we think about? Well, we have to think about, what's happening at these positions? So if we take a look at 130, at position negative 130, which is about where this is, 135. If this is 135, that's 90. So this is about that area. What we have is an enhancer region. So you don't know this yet. We haven't started looking at this, but there are points in the DNA that actually enhance the binding of the enzymes that produce the RNA that then will produce the protein. And if we're looking at renin, right, the renin protein, so this methylation of this region probably shuts down the production of renin. So you have less renin being produced, or at least makes it less likely for you to produce renin. So because it's methylated, and we looked at that methylation earlier on, this particular region becomes methylated. This enzyme, this gene is never used, or it isn't used as much. And so what you see is a severe, because of methylation, you have a less renin being made. Here you see the renin being made is different. This is less. The schizophrenic make less renin. Here you see that the methylation in this particular enhancer region is greater in the schizophrenic patient, suggesting that it's this methylation that's causing the decrease in production of renin, which again, if you think about a slide or two earlier, means that your thinking is affected. And schizophrenia is a problem with the way you perceive, the way you process the environment. And this is the reason, more likely than not. I have two again, electrophoresis gels. What looks to be radiation, I'm not sure. Yes, it looks like it's a radiation using radioactive elements bound to the particular DNA or protein. So far, we have a lot of data that really points the way to methylation impacting the production of renin, and therefore impacting the thinking in schizophrenia. people with schizophrenia. Well, here we look at reelin. This is the reelin protein, and how much reelin protein is made in the presence of the enzyme that produces methylation. What they did is they added a different concentrations in the 0, 10, 25, 50, 100, 250 of methylation inhibitor, methyl transferase inhibitor, which inhibits the methylation of the DNA. So it stops the methylation of the DNA. The more you inhibit the methylation of the DNA of that particular region that we just looked at, that increases the production of the protein. If you stop the methylation from happening, what you get at 0 means that the methylation is occurring, ready to go. At 10, it still looks like methylation. The methylation is inhibiting the production of the protein. At 25 nanomoles, you have some gene product being made, so the methylation is decreased enough that you're able to produce reelin. At a 50 nanomolar concentration, you have even more of that production. At 100 nanomotors, and then again at 250 nanomolar, you see you see a lot of the reelin being produced, because you're probably inhibiting most, if not all, of the enzyme that methylates, that DNA methyl transferase, all right, which is the enzyme that transfers the methyl group onto the DNA, because you're increasing the concentration of the inhibitor of that enzyme. So you're stopping that enzyme from transferring the methyl group. Now, they used here G3PDH as a control group, because the question that then comes, how do you know that it's this transferase isn't happening all over the place? How do you know it's specifically working on reelin? And this is the answer, because you'll notice that the inhibitor is given at the same concentration, 0, 10, 25, 50, 100, and 250, but the production of G3PDH is not impacted at all. The concentration, I think you would agree, stays about the same across all the samples, where with reelin, you get a lot more concentration of reelin if you inhibit this enzyme, suggesting that enzyme, that methylation of that particular region in the DNA, is impacting how much reelin an individual has. So it's important to understand that this is a big deal. So this particular study shows you clearly that this is at least one thing that impacts the development of schizophrenia in patients. Okay, these questions are where you're going to go ahead and earn your 100 extra credit points on this particular activity. Remember, it's going into this category, which is a lab. So it's 100 extra credit points on this lab. And you have to answer these 5 questions and show references of where you got your information. And some of this is going to be detailed, and of course you can come and get help from me, and I will help you, but I won't give you the answers. So look at questions 1, 2, 3, 4, and 5, and think about how what the answers to these questions are. We've gone through and looked at the data and kind of looked at some of what the data suggests, and I explained to you what reelin does and doesn't do here as presented in the case. But you go ahead and do your research, answer these 5 questions, and earn your extra 100 extra credit points. And good luck with this. I know it's above and beyond your high school honors biology, but I'm sure it's well within your means. I think if I will let you turn this extra credit portion in after we cover chapter 7, which is going to cover a lot of the details here, or you could turn in as early as you want, whatever you want to do. All right, then, good luck to you, and have a great weekend.